Hello guys, this is Maake Academy and I am your tutor today for Life Sciences class. My name is Fortune Maake. Guys, I would like to tell you to forgive us for right now we don't have the adequate equipment, but we do our best. As you can hear, there are some background noises, but forgive us for that. I believe before February we'll be having enough and adequate resources. So now today the topic that we are going to focus on is called DNA, the code of life. Now, I know many of us ask ourselves questions. What is DNA? Well, DNA is a nucleic acid that is constituent of chromosomes. Now, what are chromosomes? Now, chromosomes are the genetic material that we have, the genes that are responsible for how we look on the physical appearance and also our characteristics. But we are not going to focus on chromosomes because here we are going to focus on DNA. Now, DNA is a molecule. Now, this molecule is mainly found in the nucleus. Let us go here. I want us to be able to understand DNA. Now, the location of DNA, it is found within the nucleus and is called nuclear DNA because it is located within the nucleus. Together with proteins, it makes up chromosomes which turn out to go to make up the chromatin network. Now, the chromatin network, we all know the cell body. Let's say this is the cell body. And we have the nucleus here. There are some entangled material inside, which we call the chromatin network inside the nucleus. Now, in this nucleus, we find what? Our DNA. That's where we find our DNA in the nucleus. Now, it continues and says small amounts of DNA are also found outside the nucleus, extranuclear DNA, such as within the chloroplast called the chloroplastic DNA and the mitochondria called mitochondrial DNA. Now, we all know that in the cell body, we have the chloroplast, we have the cytoplasm, we have the ribosome, and we have the mitochondria inside the cell. But I'm not going to focus on mitochondrial DNA and chloroplastic DNA. Those are just extra DNAs that I just wanted you to know about. So now we're going to focus on DNA. Now, what is DNA? A DNA is a nucleic acid. Now, nucleic acids consist of nucleotides. What are nucleotides? Nucleotides, will cover them as the video goes on. We'll show you what are nucleotides and I'll explain them very in detail. Now, there are two types of nucleic acid, mainly DNA and RNA. Now, what is DNA and what is RNA? Now, DNA is deoxyribonucleic acid, which makes up the genes located on the chromosomes in the nucleus. As I told you that the DNA makes up of our genes, which are responsible of how we look. So now it says DNA is deoxyribonic nucleic acid, which makes up the genes located on the chromosomes in the nucleus. As I've told you before, that the DNA is found where? Inside the nucleus. Now we have RNA. What is RNA? Ribonucleic acid, which is formed in the nucleus but functions in the cytoplasm. This one you are going to understand it more further when we enter into protein synthesis within DNA. But right now we're just going to focus on the molecules, not on how they function and what they do. Now, where is the DNA located? Now, our DNA, as I said, it is found within the nucleus, and it's called the nuclear DNA. If you did not understand anything that I said during the part where I've spoken and covered up until now, you just pause the video, rewind it back, listen to it again so that you may understand. Okay? Yes. So, we are going to focus on the concepts that are mainly important inside DNA. We have the ribosome. The ribosome is responsible for protein synthesis. Mainly protein synthesis, we are speaking about protein production. And this protein production we are speaking about is not the proteins that build you up. It's the protein that is responsible for what? For your genes. So, these concepts may come in biological terms, or they may come in matching type questions. Those questions of 1.3, whereby you must select whether it's A only, B only, both A and B, or none. Now we have the cytoplasm, a jelly-like fluid, which is a composture of salt and water, and fills the cell and gives the cell its shape. It keeps the organelle in shape. We know we have the organelle. We know we have the organelle, which is what? Which is the cell body. Now, within the cell body, we have the nucleus. This is the nuclear membrane. Inside, we have the nucleus, which consists of chromatin network. So, outside here, we have these dots. We have this liquid called the cytoplasm, where I've put in these dots. We have a liquid called cytoplasm, and its main function is to keep the organelle in shape. Which organelle? The cell body, along with what? With what? The nucleus, the ribosome, and what? The mitochondria. Now, we continue to the third concept, the third most important concept inside this topic, the nucleus. Now, the nucleus stores hereditary material and coordinates cell activities such as protein synthesis and cell division. Now, cell division, we're going to speak about it when we enter into meiosis. But right now, we're going to speak about the DNA. As I spoke about heredity. Heredity is the genes that we inherit from our parents, or the study of genetic material. So now, the function of the nucleus is to store hereditary material. Now, what is hereditary material? It is the DNA. This chromatin network, which consists of genetic material, it is DNA. And also, it coordinates cell activities such as protein synthesis. As I spoke about protein synthesis, that we are going to speak about it in the second part of what? Of DNA. Now, let's continue. Again, I repeat, if you did not understand, you go back, rewind the video, and you listen again. And you will hear with clarification. Or you can just ask in the group, and we will attend to you quickly so that you may understand. So now, we have scientists which discovered the DNA molecule. Now, who discovered the miracle? It is Franklin and Wilkins. Rosalind Franklin and Wilkins. They were the ones who discovered the DNA molecule. These two. They discovered the molecule. Then we have those who discovered the base pairing, which is Watson and what? And Crick. We're going to speak about the base pairing as we navigate through and covering more in this topic. Now also, Watson and Crick were also given the DNA Nobel Prize. They were awarded with an award for their discovery in what? In DNA. Remember, those who discovered it is what? Is Franklin and Wilkins. Those who discovered the base pairing is what? Watson and Crick. And those who were awarded with the Nobel Prize are what? Watson and Crick. So these questions will come either in multiple choice question or will either come in matching type questions, whereby you have to choose A only, B only, both A and B, or none. These questions, they will ask you about the scientist. They will say which scientist discovered the base bearing of DNA, which scientists discovered the molecule, which scientists were credited with the DNA Nobel Prize. Then you have to know and study this scientist. You have to know this scientist on which ones played part in discovering, which ones played part in what? In discovering the molecule, the base pairing, and being credited with the Nobel Prize. Now, we're going to speak about the nucleus. The nucleus is firstly made of what? The nuclear membrane. This is the nuclear membrane. After the nuclear membrane, remember what is the function of the nuclear membrane? The function of the nuclear membrane is that it is porous. When we speak about something that is porous, we are speaking about something that has holes in it. It has spaces within it, small spaces which allow for something to pass through. When we speak about porous. So you see these spaces here. These spaces between, these are pores. Pores of the DNA membrane, which allow something to pass through. Okay? To pass through. So, now we have what we also call the nucleoplasm. Now, the nucleoplasm is a jelly-like liquid that fills the nucleus. Mainly, you'll see it elaborated as dots inside what? The nucleus. Now we have the nucleolus. Now this nucleoplasm, let's go back to the nucleoplasm. This nucleoplasm consists of nucleotides. Nucleotides which are roaming freely in the nucleoplasm. Hear me again. I repeat this. The nucleoplasm, the jelly-like liquid that fills the nucleus, consists of free nucleotides inside of it, which are essential for DNA replication and which are essential for what? For RNA formation. I hope you heard me. The nucleoplasm consists of free nucleotides. Nucleotides. And which are essential for what? For RNA and DNA replication. I hope you understand. And I hope you heard me. If you didn't understand, you go back, rewind the video so that you can understand. So we also have the nucleolus, which produces RNA and ribosomes. We also have the chromatid network. As I told you before, that within the nucleus, we have entangled mass of hereditary material. This entangled mass, it's called chromatin network, and it's a mass of genetic material composed of DNA and proteins that form what? Chromosomes. As I told you, if you want to understand about chromosomes, you'll understand them as we further enter into what? Into meiosis. For now, we are still focusing on what? On DNA. Okay? Now, we're gonna know about this nucleic acid. What are nucleic acids? Nucleic acids, they control protein synthesis. As I spoke before, I said protein synthesis, it is the what? The production of what? Of protein. Protein synthesis is what? The production of proteins. And it takes place in 3 stages. And those 3 stages, we will discuss them in the second part of DNA. Now, there are 2 types of nucleic acids. As we spoke before, we know our 2 types of nucleic acids. We have DNA and we have what? RNA. Now, all of our nucleic acids consist of what? Nucleotides. Now, the examiner might not say nucleotides. Sometimes, or other books or question papers or other teachers may not use the word nucleotides, but they may use this word called monomer. Monomers. Now, what are monomers or nucleotides? They are the building blocks of what? Of the DNA molecule. They are the building blocks of what? The molecules. Building blocks consisting of what? Sugar, nitrogenous base, and what? And phosphate. So now, here is the structure of a nucleotide. Now, the nucleotide, as I spoken, it is a building block consisting of what? Of sugar. Consists of sugar. Consists of nitrogenous base, and it consists of what? Phosphate group. Now, this is a building block of a what? Of a nuclear, of a DNA, or of a molecule. So now, the nucleotide consists of a phosphate group, the pentose sugar, and the nitrogenous base. This is a nucleotide. When it is in many, in plural form, it's called nucleotides. You can either call it a nucleotide, a monomer. In plural form, you can either say nucleotides or monomers. Even in the exam question paper, the examiner will tell you will use different concepts. Sometimes he might use monomers, sometimes he might use nucleotides. It depends on the examiner. So this is the structure of what? Of a nucleotides. Make sure you keep it in record. Now, we are going to enter into the DNA molecule. Now, we have DNA, which is deoxyribonucleic acid. Now, there are things, these things that I tell you here, they're going to ask you in the exam question paper or in your question paper. The examiner is going to ask you. Now, the DNA has a double helix shape. A double helix shape. And it is what? Double-stranded. Here, I shouldn't have used the double-stranded. It has a double-stranded structure. Now let me edit it. Double-stranded structure. Yes, the DNA molecule has a double-stranded shape. A double helix shape. A double helix shape. And in structure, it is what? Double-stranded. Now, it looks like a ladder when it is uncoiled. Now, let us go to the structure of the DNA. What does the DNA looks like? Now, when it is coiled, it looks like this DNA structure, which we all know, which we are all familiar with. We know that when they are speaking about DNA, we expect something like this. But this is the shape, the double helix shape of the DNA. This is the double helix shape of the DNA. This one is the double helix shape of the DNA. But however, this one is the structure. Because as you can see, we have the first strand. Forgive me for that. We have the first strand here. And we have the second strand here. As I said, it has a double-stranded shape. We have the first strand here, and we have the second strand here. That's why it is double-stranded in shape. And remember when I said nucleotides are building blocks of what? Of nitrogenous bases of DNA. They consist of phosphate group, pentose sugar, and what? Nitrogenous base. This is our what? Our nucleotide. First nucleotide, second nucleotide, third nucleotide, and fourth nucleotide. You see, they are building blocks. They build up a strand. Even this side also, first nucleotide, second nucleotide, third nucleotide, and fourth nucleotide. They build up a strand. They are the building blocks of what? Of the DNA molecule. Now we have the components of a DNA molecule. What are the components of a DNA molecule? The components of a DNA molecule are what? Nitrogenous bases with weak hydrogen bonds. Now what are weak hydrogen bonds? Weak hydrogen bonds are these bonds. You see these blue lines here. These blue lines. They are hydrogen bonds which connect nitrogenous bases. Our nitrogenous bases are going to be here, are going to be paired here. They are paired and joined together by these weak hydrogen bonds. Okay? Now, another component, it is the sugar. Now, the sugar of a DNA molecule, this is not the sugar that we eat at home. This is the sugar that is needed for your body to begin to build up proteins. And it is produced within the DNA. Now, the sugar that will stand here in the DNA molecule, it's called deoxyribose. Hear me very well. The sugar within the DNA molecule is what? Deoxyribose. Is the sugar within the DNA. I hope you understand. So now we do have nitrogenous bases, and our nitrogenous bases, they do vary. Wait for me to clear these red lines. Our nitrogenous bases, they do vary. We have guanine, we have cytosine, we have adenine, and we have thymine. Now, how do these nitrogenous bases work? Now, I'm going to explain them, these nitrogenous bases. Cytosine goes together with guanine. Adenine goes together with who? With thymine. Same again with guanine, it goes together with cytosine. And the thymine with what? Adenine. So these nitrogenous bases are complementary to each other. When I mean they are complementary to each other, I mean guanine pairs with adenine, and I mean adenine pairs with thymine, and cytosine pairs with guanine. They are pairs. So guanine and cytosine, we call them the good couple because we call them the good couple. Good couple. Why do we call them the good couple? Because they are always paired together in both DNA and RNA. But however, adenine and thymine are only paired together in what? In RNA, because in DNA we have thymine, but in RNA we have uracil. So there is no thymine inside of what? DNA. Immediately when we don't have thymine inside of RNA, in DNA we have thymine, but in RNA we don't have thymine, but we have instead uracil on the place of thymine. So therefore in RNA it is uracil pairing with adenine, but in DNA we have adenine pairing with what? With thymine. I repeat, in DNA we have thymine, and in RNA we have uracil. And in DNA, thymine pairs with adenine, and in RNA, adenine pairs with uracil. Now, I hope you pause the video, copy the structure of DNA so you can know that the shape, when we speak about shape, it is only when it is coiled. When we speak about the double-stranded shape, it is when it is uncoiled. Okay? Yes. If you did not understand anything that I said, you pause the video, you rewind so that you can understand. So now we go to RNA. Now what is RNA? RNA is ribonucleic acid. It has a single-stranded structure, not shape. Let me edit it. Has a single-stranded structure. It doesn't have a shape. It only has a single-stranded structure. So what are its components? The components of RNA is sugar. We have the ribose sugar. Remember, in DNA we had what? Deoxyribose sugar. Now they will ask you, which sugar do we find in DNA? You will then say it is deoxyribose. But when coming to RNA, they will ask you what kind of sugar is in RNA. Then you will say ribose sugar. Now the sugars in RNA and DNA are what? Are different. They are not what? They are not the same. Now we have the nitrogenous bases here in adenine, in RNA. The nitrogenous bases in RNA are adenine, uracil, cytosine, and guanine. Remember, in DNA we had thymine in the place of what? Of uracil. But now in RNA we have uracil in place of what? Of adenine, of thymine. So here we expect adenine to pair with who? With uracil. cytosine to pair with who? With guanine, because cytosine and guanine are the good couple, so they're always paired together. But adenine and uracil are paired together only in RNA, and in DNA we have thymine pairing with adenine. I hope you understood how far we have reached now with this topic. Now, that is the basic description of the molecules. Okay? Now let us go to DNA replication. So now, before we go to DNA replication, I want us first to understand the similarities between DNA and RNA. Both DNA and RNA contain sugar alternating with phosphate. Remember the nucleotide structure. Remember the nucleotide structure. We have our phosphate group here, and we have our sugar here. So both of them, they have alternating what? Sugar and phosphate. Now, when we mean alternating, we mean things that are attached to each other, things that rotate around each other, things are close to each other, things that are exchanging to each other. Okay? Now, both of them have nitrogenous bases, adenine, guanine, and cytosine. Remember, DNA share the similarities that they have. Both have what? Adenine, guanine, and cytosine. Why is not thymine and uracil not included? Remember, uracil is only found in RNA. Uracil is only found in RNA because it pairs with adenine in RNA. But however, in DNA, we have thymine. In DNA, we have thymine, which pairs with what? With adenine inside of the DNA molecule. Remember, in the RNA molecule, we have what? We have uracil, but in the thymine molecule, in the DNA molecule, we have the thymine. Now, both of them, they share thymine. They all share the similarities of sharing adenine, guanine, and cytosine. Now, what is another similarity? The third similarity is that they both play a role in what? In protein synthesis. Both DNA and RNA play a role in what? In protein synthesis. Now, as I said, you're going to understand protein synthesis when we make another lesson, when we cover part 2 of DNA. Okay? Now, let us go to the differences between DNA and RNA. Remember, these questions the examiner is going to ask you in the exam. And he's going to say tabulate 2 differences or 3 differences or 4 differences between DNA and RNA. Remember, you have a mark for the table, 2 marks for the first difference, 2 marks for the second difference. So it's going to be 5 marks. 5 marks. This question mostly come in 5 marks. Sometimes the examiner might say 4 marks or 3 marks. 1 mark for the table and 2 marks for the difference. Might say tabulate 1 difference or tabulate 2 differences, depending on the examiner. Now, you must know these differences between DNA and RNA. Now, DNA is found within what? The nucleus, chloroplast, and what? Mitochondria. However, DNA is found in the nucleus, chloroplast, and mitochondria. However, the RNA is found in the nucleus, and the cytoplasm only. But DNA is also found where? In mitochondria. This one is only found in the nucleus and cytoplasm. I'm referring to RNA. Now, the DNA is a double-stranded molecule. It has a double-stranded what? Structure. Whereas the RNA has a single-stranded structure. It's a single-stranded molecule, the RNA. And DNA is a double-stranded molecule. Now, what other difference is there? DNA contains the sugar deoxyribose, whereas RNA contains the sugar ribose. Remember, I emphasize on this again. DNA consists of the sugar deoxyribose and RNA consists of the sugar ribose. Now, they're going to ask you in the multiple choice, or they're going to ask you in the matching type question, and they're going to say which sugar is found in RNA, and they're going to say you're going to have to choose between A only, B only, both A and B, or none. They're going to give you options. Or if you find it in multiple choice, they're going to give you A, B, C, and D. Then you're going to choose one, which is right. You're going to choose the one which is right. Now, which nitrogenous base does they have? What is another difference? Another difference is that DNA consists of the nitrogenous base thymine, whereas RNA consists of the nitrogenous base uracil. Remember, I told you that in DNA we have thymine, but in RNA we have uracil. This uracil pairs with adenine in what? In RNA, but thymine pairs with adenine where? In DNA only. I hope you understood. And if you did not understand, you rewind the video. You listen to the you listen again, and you will understand. And if you find difficulties to understand, please mention your difficulties in the group so that we may attend to it rapidly. Now we are done with the similarities and what? And differences between RNA and DNA. Now we're going to focus on DNA replication. DNA replication. I think it cancelled my... So this is DNA replication. So this is DNA what? Replication. Now, what is DNA replication? DNA replication, it is when a DNA molecule makes an exact copy of itself. Now, it means the DNA will make a copy of itself, an identical copy of itself, something that looks like it. It will make another DNA which looks like it. That's why we say it is when a DNA molecule makes an exact copy of what? Of itself. Now, what is the significance or importance? Remember, when we speak about significance, we are speaking about what? Importance. Now, DNA replication ensures that each daughter cell gets an identical copy of the original DNA after what? Mitosis. Now, we all remember mitosis in grade 11, that it is where it is the process whereby it is the cell division whereby a cell makes an exact copy of itself, right? We know that it is the type of cell division whereby a gamete will result, a cell will result in two. It's a cell division whereby two genetically identical cells are produced. But I don't want to focus on mitosis because we have not yet reached at meiosis. I'll explain mitosis when we reach meiosis. Now, what is another significance of DNA replication? Remember, you must copy this down because They're going to ask you in the exam. They're going to ask you in your question paper, and you must know them. Now, the significance and importance of DNA replication is that another significance is that it ensures that each daughter cell gets the correct number of chromosomes after meiosis. So as I spoke again, for you to come and understand these significances, it's going to need us to enter into meiosis, but we're going to cover that when we reach into meiosis, so that you may understand this too, why we speak about chromosomes after meiosis, the identical copy, things like that. But we're going to understand them deeper when we enter into meiosis. Now we're just giving you a brief, okay? But for now, just know them by heart, because you are still going to understand them further as we enter into meiosis, the topic of meiosis, because right now we're still in DNA, we still have protein synthesis to cover. Now, we have the process of DNA. Now, the process of DNA replication, you know, they give it 5 marks, 6 marks, or 7 marks. So they're going to ask you in the question paper to describe the process. So this process, you must know it by heart. So we hear the first point, it says the DNA double strand does what? It unwinds. As you can see here, it was winding. Now it is unwinding. It is becoming uncoiled. Here it was coiled. Here it is becoming uncoiled. Okay? The DNA double strand unwinds. It was winded here. Now it is unwinding. Now the double helix unzips. You can see it is unzipping. It is separating. We know that if you have a jersey and you are unzipping your jersey, we know that you are separating the hemispheres of the jersey. You are separating the left hemisphere with the right hemisphere so that they can be room for ventilation. Now, here the DNA double helix does what? It unwinds. It unzips. Now, weak hydrogen bonds between nitrogenous bases break. You can see that here our nitrogenous bases were joined together by what? Weak hydrogen bonds. They were joined together by what? Weak hydrogen bonds. But now the weak hydrogen bonds begin to separate, begin to break, because the double helix is what? Is unzipping. Then it results in 2 separate strands. We can see we have 2 strands. We have the first strand. Wait for me. I'm still removing so that we can understand. We can see that it says what? It results in 2 what? Separate strands. Now we can see that we have the first strand here and the second strand here. After what? After unzipping. So each strand is then used as what? As a template to form a complementary DNA strand. Now you can see what? That it is forming a complementary DNA strand. You see? A complementary DNA strand. Complementary DNA strand. Using what? Free nucleotides from the what? From the nucleoplasm. As I stated before, that the nucleoplasm consists of what? Of free nucleotides. Those nucleotides consist, those free nucleotides can either be RNA nucleotides or free DNA what? Nucleotides. Now, the process says what? Using free DNA nucleotides. Make sure that when you describe this process, make sure that you indicate that these nucleotides are DNA nucleotides. Now, using free nucleotides to form what? To form 2 genetically identical DNA molecules are formed. You can see that now we have 2 genetically identical DNA strands. We have new strands. We have new strands, new DNA molecules. which are identical, which are genetically identical. You can see here that adenine is pairing with thymine, cytosine is pairing with guanine, thymine is pairing with adenine, so on and so on. Even on the other molecule, still the same thing. Even the base pairing is still complementary to what? To the original DNA strand. Remember, it says to form a complementary DNA strand. Remember the original DNA, guanine paired with cytosine, thymine paired with what? With adenine. Even in the two genetical identical molecules which are formed at the end of the process, still contain of the same base pairing. Okay? Now, this process is controlled by enzymes. Make sure that you don't forget to include that also when you describe the process. Now, I'm going to restart explaining it again. Now, I say the DNA double helix unwinds. You can see it is unwinding here. The double helix unzips. You can see here it is unzipping. Weak hydrogen bonds between the nitrogenous break. Why do these weak hydrogen bonds break between the nitrogenous bases? Because the DNA double helix did what? Unzipped. Resulting in what? Two separate strands. We can see these two separate strands. And each strand is used as a template. You can see it consists of what? Of nitrogenous bases. And using free nucleotides from the what? To form a complementary DNA strand. Using free nucleotides from the nucleoplasm. And I repeat, the nucleoplasm consists of free nucleotides. The nucleotides may be RNA nucleotides or DNA nucleotides. Keep that in mind. Make sure that when you describe, you emphasize on the nucleotides that they are DNA nucleotides to form two genetically identical molecules. Two genetically identical DNA molecules are formed, and this process is controlled by what? By enzymes. Make sure that you note this process down. Make sure that you note this process down. Make sure that you note it down, because the examiner is going to ask you in the exam about this process. So if you want to note it down, you pause the video and you note it. After noting it, you continue with the what? With the video. So now we are finished with what? With DNA replication. And now we're going to go to the last part of the introduction of the DNA molecule and RNA molecule, which is what? DNA profiling. Now, what is a DNA profile? A DNA profile are patterns or black bars on X-ray film when an extract of DNA is put through a special biotechnical process. Now, what is DNA profiling? DNA profiling is a method of identifying an individual by comparing his or her DNA profile with another unknown DNA profile. Now, DNA profiling, it's mainly used to help us identify. Now, when we speak about DNA, when we say we're going to go for a DNA test, this is what we are going to look for. They are going to check these bars, whether they match the bars of the one that they are testing for. You see here it says semen and it says victim. It says suspect number 1, suspect number 2, suspect number 3. You know, it shows of let's read the scenario. It says study the diagram below, which shows the following DNA profiles or genetic fingerprints: blood of a raped victim, blood of the 3 suspects, and semen found on the female. Now, this DNA profiling helps us to identify individuals using another unknown DNA profile. Let's say we are looking for the father of the child. You don't know the father of your child, and you have 3 people that you are thinking of. They are going to take their DNA, take the DNA of the child and take your DNA, and they are going to check whether the DNA of the child matches your DNA, matches which DNA of those men. First, they are going to extract the DNA of the mother. After extracting the DNA of the mother, they are going to extract the DNA of the victims, of the suspect. Okay? If you don't understand, you're gonna ask it is highly recommended for you to ask in the group or to rewind the video and listen to what I said. They are gonna first extract the bars of the mother away from the semen and see how many bars are left there. So after they've identified how many bars are left there, they are now going to be able to identify which one of these three suspects is responsible for this semen. I hope you understood. Now, let's go back. What is a DNA profile? A DNA profile. A DNA profile are patterns of black bars left on an X-ray film when an extract of DNA is put through a special biotechnical process. Now, what is DNA profiling? DNA profiling is a method of identifying an individual by comparing his or her DNA profile with another unknown DNA profile. Now, what are DNA profile? What are the uses of DNA profile? Now, DNA profiles are used for the diagnosis of inherited disorder such as cystic fibrosis, hemophilia, sickle cell anemia. So mainly the DNA profiles can be used to check where you inherited a certain disease. Let's say you are having a certain sickness and you go to the hospital. When they take your blood test, they are going to check where you inherited the disease, whether the disease just developed within you or whether you took it from your mother, your father, or your grandmother or something in the family. Because our grandmothers, they pass the genes to our mothers pass the genes to us. So they are gonna check for that. Now also it is used to identify criminals in forensic science. Now when it comes to crime scenes, you find that someone is shot and the blood falls on the ground, they are gonna check the forensic site for DNA, fingerprints, stuff like that, so that they can be able to identify the criminals which were part of that crime scene. Now, it is also used to identify relatives, such as establishing whether a particular person is the father of a child or not. Remember, if you are not, your mother doesn't know your father, or you are a woman, you don't know the father of your child, and you've slept with many men and you can identify the father. They're gonna take those 3 men, they're gonna take the DNA of the child, they're gonna take your DNA, they're gonna extract your bars out of the child, then the remaining bars, they're gonna match them with the bars of the fathers to see which one is the father. Now, tracing siblings, brothers or sisters who have been separated by birth. In hospitals, we know they can steal a child. Let's say you gave birth to twins, then they separate the other one, they steal the other child. Then you end up meeting in the future. You end up finding each other in the future. So they're going to use DNA profiling to see whether you are siblings or not. Now, it is also used to identify people who have died where their bodies cannot be recognized. Let's say you have someone in the family whom died during when the house was burning, or maybe who died during a car accident which was brutal, or maybe they died and they decomposed and we can no longer identify them. So they're going to take your DNA in the family and they take that person's DNA and check whether this person is from your family or not, because they are unrecognizable anymore. So the only method is to use DNA profiling to check whether they are of your family. Now, what are some surrounding debates around DNA profiling? You know, DNA profiling can, when we're saying debates, we're speaking about things that hinder the validity of the DNA replication, whether DNA replication can be trusted or whether it cannot be trusted. So now, DNA profiling is generally accepted as being extremely reliable. However, DNA testing sometimes can be controversial. Now, DNA replication is reliable. It can be trusted. It has a guarantee that it can bring results, good results. But the testing, when in the lab, when they are conducting the test, there can be controversy. There can be some mismanagement of the testing in there. Now, why? Since only a small piece of DNA is analyzed, a DNA profile may not be unique to an individual. Now, this basically means that since a small portion of DNA is analyzed, the DNA profile might not be accurate to a certain individual. So it will need a broad spectrum. It will need them to take a big portion of DNA. So DNA profiling is performed DNA profiling performed in private laboratories may not follow uniform testing standards and quality controls. So because it is tested in private labs, you might find that the person in the lab may not follow the procedures of doing DNA replication correctly. So he ends up making the profile test without following the measures that are needed for the profile to be accurate. So it will end up giving us unreliable result. So another reason is that since human beings must interpret the test, human error could lead to false result. Now, because we are humans and we know we have errors, you might find that we are doing testings and we take the wrong samples. Or let's say we have 4 DNA profilings that we have to make. There are 4 cases that we have to make DNA test for them. Then you end up mixing the cases. You take the DNA profile of someone who was raped. You put it to a DNA profile of someone who wants to identify the father of the child. So those kind of errors of mixing samples, of mixing the wrong samples can lead to the results being false. Also, another reason is that DNA profiling is expensive. Yeah, we all know in South Africa we are mostly from disadvantaged backgrounds, so DNA profiling is expensive. So another thing is that DNA analysis might reveal personal information such as the presence of HIV and AIDS. This could be used against the person because of prejudice against the persons with disease. So mainly DNA profiling, let's say you raped someone, I'm just assuming. Then you have AIDS, HIV and AIDS. So when they're going to present your DNA in the court, they're going to actually mention that you have AIDS, and we all know the kind of discrimination and judgment people with AIDS, HIV and AIDS in South Africa they go through. So this is another disadvantage because people's secrets, personal diseases will be revealed to the public, and which is not good. That everyone must know that you are suffering from a certain disease because it is your own disease and it is your privacy. So DNA profiling ends up bringing an invasion of privacy. You see? So now, if you did not understand or you want to copy down this, you just pause the video, you copy, then you continue with the video. Same as the uses of DNA profile, you pause the video, copy, and you do what? You continue with the video. You pause the video, you copy, then you continue the video. So now we're gonna study this diagram and I'm gonna show you how to interpret this diagram of a DNA profiling. Now here we have a woman who's raped. Here is the victim. and these are the 3 suspects. Now they want to know who raped her. So first we can see that this sperm has this bar. First bar has the second bar, has the 4th bar, the 5th bar, the 6th bar, and the last bar. So obviously the semen cannot have the woman's DNA inside. It has to have the man's DNA, or it might have the woman's DNA. So now we're gonna check which man matches this semen. The first suspect doesn't have this big bar. He doesn't have this second big bar with the small lined big bar. Even the second suspect also. But the third suspect has what? The big bar and this second one. This one's match. Also, the semen has these 3 bars here. This one doesn't have it. Suspect number 2 doesn't have it, but suspect number 3 does. We check the semen again. Suspect number what? Number 3 still has what? This bar here, which corresponds with this one. But this one does not correspond. This one and this one does not correspond with this bar here. And lastly, suspect number 3 has this bar, which correspond with this one. But suspect number 2 and 1, they don't have bars which correspond with this one. So suspect number 3 here is the one who raped this victim. Let me repeat again. And this time I'm gonna be a bit slow. So we're gonna check for whose semen is this. Remember, semen is a mixture of sperm cell and fluid. So we wanna check who raped this woman. So first suspect doesn't have this big bar. Here doesn't have this. Here doesn't have this. Here doesn't have this. And here he doesn't have this either. Second suspect also doesn't match with this one. But the third suspect, the first 3 bars, they do match. Second 3 bars, they do match. The last 2, 3 bars, they do match. So meaning that the victim is raped by suspect number what? Number 3. So I'm gonna send activities in the group pertaining the videos that pertaining the content that we covered today. I'm gonna send one activity for DNA replication, one activity for the molecules, one activity for DNA profiling. Okay, thank you guys. This was a very wonderful lesson. This is Maake Academy, and I am your leading life science tutor, Fortune Maake, and I thank you for attending this class. Peace.